Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Patrick Gurgel from Prometic Bioseparations.
Thanks, Leah. Hello, everyone. Well, good morning, good afternoon, or good evening, depending on where you are in the world. As Leah said, my name is Patrick Gurgel. I'm the head of research and development of Prometic Bioseparations. We are a company in Cambridge, in the UK. And I'm going to talk to you a bit today about the work we do on the development of Affinity Chromatography Adsorbents for bioseparations. It's a very short talk, so I just want to give very briefly. I think most people have heard this, but what are the bases of affinity chromatography? As you probably heard, affinity chromatography is just a method of separating different compounds based on specific interactions between the compounds or the target that you're looking for and a ligand immobilized onto a base matrix. Those interactions typically in affinity chromatography are multimodal in nature, so there's not a specific force that drives that, but most likely a combination of forces. It's very common to have different possibilities as ligands, such as proteins, antibodies, receptors, and inhibitors for a specific target. Metals, also peptides and small chemical compounds. The last two, especially the small chemical compounds, are the specialty of Prometic. We specialize in developing those kinds of ligands for specific targets. As affinity chromatography ligands, they have some particularities that would be at least very desirable, and they need to be chemically stable. Of course, you want to bind and elute your target from them, so that's an important thing. And you want to use mild conditions because typically you are targeting something like a protein that has some kind of biological activity, so you want to preserve that. You want your ligand to display low toxicity in case of leaching, of course, and of course be relatively inexpensive. So what is the biggest drive of affinity chromatography? Typically the people look into affinity chromatography because you have the ability to resolve complex problems in bioseparations using fewer steps than if you use a non-specific chromatographic or other steps. So on the left-hand side in that diagram, you can see a comparison of, in red, a process with multiple steps, and each step is losing about 5% of your target. And you can see after multiple steps, at the very end, your yield is drastically diminished. In contrast, in blue, with affinity chromatography, you can see that ideally in one step, you resolve your mixture, and then you get a much better yield. So the right-hand panel shows an ideal situation of what we can achieve with affinity chromatography. This example is putting human plasma, which is in lane 2 in that figure, through one of our mimetic blue adsorbents This is an adsorbent for albumin. And you can see in lane 3, this is the flow-through. And in lane 4 is the elution fraction with albumin. You can see a small band down at low molecular weight too. But you can see that with one step, essentially, you remove pretty much all the contaminants, and you've been able to recover albumin. So this is the ideal situation. The thing that happens a lot with affinity chromatography that people many times just don't think about or forget if they know is that if you want to develop a new affinity adsorbent, and that's very much the case for a lot of target proteins that people come to talk to us about. If you want to develop that, you need to develop most of the times a new affinity ligand. So that requires quite a bit of effort and time, and of course, as any research program carries some degree of uncertainty. One thing that is problematic in downstream in general, it's changing but still somewhat problematic, is that many times people are so involved in expressing and producing the protein of interest that the downstream processing steps become secondary, and they don't spend the time that they should during the development process to develop something. And even in that case, in that scenario, affinity chromatography many times is an afterthought, that people will try several other techniques first and then come back to affinity chromatography if it didn't work very well. We have developed a few tools that make this process a bit easier, and I will talk a bit more about this in a few slides. But in general, just to give you an idea of what's the process of developing an adsorbent for affinity chromatography from scratch, this is summarizing this figure that you can see now. So we start with the ligand discovery step, which then I'll give more details on each one of the steps in a minute, but just in general. We start with a ligand discovery, which prompts typically a milestone. Then we do the activities about adsorbent development and chromatography development. And the distinction between those two is that in adsorbent development, we are dealing more about the chemistry of producing the adsorbent itself. And in chromatography development, we are talking about the process of using that adsorbent. Those are done relatively in parallel, and they trigger another milestone upon completion, which then lets us do the tech transfer from the research scale that we do here in Cambridge to our production facility, and also do process validation for making the adsorbent. And at the same time, we work on the regulatory support package and activities. After that, you should have a fully validated adsorbent that can be used in your process. So just to give a bit more detail on each one of those phases, as you can see here, we divide into the 4 phases. The first one, the ligand discovery, is the one that we have developed a handy tool to facilitate or remove some of the work from our hands to the clients or your hands. So in phase 1, when we do ligand discovery, we can have different activities such as ligand design, the library synthesis, of course, and library screening, which then once you identify a ligand that you are interested in, we have a verification chromatography step. If you want, we can do that in-house as part of development process, but one possibility is for the client to use what we call the MMM plates. And MMM plates stand for multi-mode mimetic ligand plates. Those are small libraries, 96 well plates essentially. So they are micro columns, about 250 microliters of each one of the adsorbents that cover a relatively vast chemical space. We have a variety of hydrophobicities and charges combined in ligands that have then final multimode characteristics. You can see on the top left panel somewhat of the map of the different areas that we have in our ligand. So if you acquire one of those plates, you can then use at your own time. You don't have to disclose to us what is the protein of interest or transfer any of the methodologies that are needed for screening. So it makes the process faster in some respects, and you can do as many times as you want under as many conditions as you want. And then you would come back to us saying, hey, this specific ligand or this column or this row is working well. Just to give you an idea of how well it works, on the left, on the bottom panel, the SES page image there, it's just all the proteins from plasma, human plasma in this case, that bound to each one, to several of the adsorbents that are on that plate. And you can see that the profile of bound proteins changes a lot from one to the other. This is to demonstrate that there is pretty much a ligand for any kind of protein that you want. On the right here are a few examples of actual screenings for different plasma proteins using full plasma. And you can see that those are heat maps. So essentially the red, the ones marked in red are the ones that display good binding. So you would come back to us and say, well, my protein of interest bound best to column 6. And we would start a conversation about maybe developing one of the adsorbents in that column or something derived from that column. So this essentially makes phase 1 a bit more flexible and can be started maybe earlier in the process than you guys would be ready to engage on a proper development program with Prometic. But once you find something and we do the verification of chromatography and we're all happy with the preliminary results, then we move on to the phase 2, which is the adsorbent and process development. So in terms of the adsorbent development, our choice of base matrix is typically agarose. Is something that we do in-house, and it's stable, it's safe, it's relatively inexpensive, and it's very versatile in terms of uses. But there are other natural polymers and synthetic polymers that can also be used, that we have used in the past. We do work on determining the optimal ligand density for a given adsorbent. More of the ligand is not always the best solution. You have to get a balance there, because more ligand many times is connected to higher non-specific binding. We work on attachment chemistries and the use of spacer arms, and also optimizing the actual synthesis method for the ligand itself and for the adsorbent. As I said, at the same time in phase 2, we do work on the performance assessment for the adsorbent. What are the load, elution, and wash conditions that you would be likely to use, as well as CIP conditions, which are very applicable to your process, and also run reuse studies for the adsorbent. We then move on to phase 3, when we are all happy with the way the adsorbent is made, the process is in control, and the yields and purities hit the mark that the client needs. And then at that phase, we work on producing verification batches of the adsorbent. Typically, We have 3 of those made at small scale in our research facilities. Then we have an additional tech transfer batch, which is typically done at an intermediate scale between the bench and the proper production scale. And finally, we produce 3 validation batches that are typically run at production scale, but can be run at smaller scale. And at that point, the validation batches are typically already used by the client for either for preliminary processing studies or even in campaigns in some cases. So in conjunction with that, we have also phase 4, which are the activities that more need some of the material that is produced in phase 3 to really work. And in this, we include the extractables and leachables studies for the final version of the adsorbent, as well as the stability of the material, the shelf life, and the different conditions. And if needed, depending on what the nature of the ligand is, a toxicological evaluation of the adsorbent and the ligand in some respects. So I hope that in this very short presentation I gave you a general idea of what happens when we are developing an adsorbent for affinity chromatography. And just as take-home message, I want you to remember that affinity chromatography is a powerful tool for bioprocessing. It has a history of success in this field. It does, for when you want to apply that, require some planning and some development and some effort to be put on that. That we have tools available to make the whole process a bit faster and more flexible that you might want to take advantage of. And finally, that we have been working on this on affinity chromatography for about 30 years, and that we're here to help. So with that, I'll open the floor for some questions. Thank you very much.
Okay, great, thanks. So the first question is, if the mimetic blue has a superchron blue as an affinity ligand, how different are these beads from other beads on the market?
Well, the mimetic blue is not exactly like superchron blue, but the great advantage of our material also is that our agarose beads are nearly monodispersed. So they are in size, they are very much similar to each other, and that makes the flow properties very predictable and very good. So that would be, even if the ligand is the same, the performance of the adsorbent most of the times, it's beneficial to have this nearly monodispersed material.
And what happens if we get a hit on the screening plate?
So if you get a hit on the screening plate and you come back to us and tell us which one that is, and you want to engage on a development program, there are two ways to proceed. One, maybe that hit itself is already good enough, and we would do the verification chromatography just to ascertain that, at least preliminarily. Or if it's not, if you have a group of ligands that seem to be good, we can expand the library and concentrating in that chemical space. So produce a few more ligands that have similar characteristics to that one and test those. So it becomes an iterative process. And typically with 2 rounds of that, we get a ligand that works well enough for most cases.
Okay. And how do you determine the right matrix for your ligand?
So essentially most of the times is test. We test everything. So, but your process might have some constraints that would prevent using this matrix or that. For instance, you might need to use NaOH to clean your material, which then rules out a few base matrices. But in our case, we will always start with either, most likely with agarose and polymethacrylate, which we know work very well, but there are other possibilities that can work, but you have to test, essentially, unless you have a specific constraint that rules out a base matrix or the other.
Okay, and this will be the final question. If we didn't get to your questions, all your questions typed in the chat window will be passed to Patrick, and he can follow up individually with you following this. So the last question we're going to do for the webcast, though, is how many custom resins have been, or have any custom resins been used in an approved product manufacturer?
So right now, I think we have 18 processes that have been FDA or EMA approved using Prometic adsorbents. So, yeah, 18.
Okay. So thanks, Patrick.
Oh, no, thank you all.
And
And
thanks to
thanks,
our
Leah.
audience for yes, and thanks to the audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Goodbye.